So, Daktari, welcome.
Good morning, ladies and gentlemen. Welcome to our last day of this session. And I would like us to start with a word of prayer. It's always good to humble ourselves before the Lord because it is in Him who has made this thing possible. Let's pray.
Recording in progress.
Our Heavenly Father, in the mighty name of Jesus Christ, we come before your throne of grace and mercy this morning with thanksgiving in our hearts. Thank you, King, for glory, for that you are taking care of us, O Jehovah. From Tuesday, Jehovah Father, up to this today, Friday, King of all glory. Indeed, we have seen your goodness and your mercies upon our lives, O King, for glory. We are blessed, Lord, O Father, to be in your presence, O King of all glory, even learning things that we have never heard, O King of all glory. Some of them we have heard from books, O Jehovah Father, mighty and everlasting Father. We have talked to people, O King of all glory, who have brought their personal experiences, their farms, O Jehovah Father, and we have seen indeed what you have done, O King of all glory. Jehovah Father, we thank you, King of all glory, because in these technologies, O Jehovah, you had it before the foundation of the earth, because you planted Adam and Eve in the garden, O King of all glory, to do it, O Jehovah Father. All what we are doing, Jehovah Father, is to follow you, Jehovah, and to do what you intended us to do, King of all glory. Thank you, Heavenly Father, eternal everlasting Father. We thank you for every speaker, Jehovah Father. From Tuesday, Jehovah Father, we have had various speakers, O Jehovah Father. Strengthen our minds, O Jehovah Father, King of all glory. Try to focus things which will help your people, O Lord Almighty. And indeed, Jehovah Father, if you follow these rules, Jehovah Father, indeed, Kenya will be different, O Lord Almighty. And your people will have food to eat, O Jehovah. Even our animals, O King of all glory, they will have something to eat, O Lord Almighty. We thank you, Jehovah Father, for that knowledge, eternal everlasting Father. Even this day, Jehovah Father, as we start our session, Lord Almighty, how we pray, Lord Almighty, you are anointing upon the speakers, Jehovah Father, those who speak to us, O King of all glory. May you anoint their ones, O King of all glory, that whatever they reach, Jehovah Father, we come in the clarity, O Father, in our mind, O Jehovah Father, and we shall be able to catch up, O Jehovah, what they are teaching us, O King of all glory, and we shall do us of these things, O Jehovah, to the glory and honor of your holy name. We thank you and we praise you in Jesus' mighty name. We pray, we believe and trust. Amen. Thank you. Ladies and gentlemen, I don't know whether our speaker is ready to come and introduce to us the issue of strategic for conservation of crop genetic resources. Has he really arrived? Not yet. Anyway, I would even before he comes and give my personal experience, this conference is that personally I have benefited. Basically, I am pharmacist by training, more so in clinical pharmacology. And when Dr. Gu touched on synthetic biology, now it reflected back to my mind how this is in the modern medicine can be possible, and I know it is being done. Only that it has not much talked about, because when we talk about IVF, in fact, we are talking about synthetic biology. That is what is happening, actually. Only that it has not been very much being utilized. And I thank the organizers of this conference because of what they have brought to us and how they have opened our minds, that back in my village where I come from, I'm able to talk to people. There is a modern way of farming these days, not the traditional ways of repeating the same crops, the same place, and such and such. So it is good for each of us, wherever we come, if we can take this gospel of food production. Indeed, Kenya can no longer talk of hunger. Kenya can no longer say we are going to import food from other countries. Indeed, even our neighbors, we can be able to feed even our neighbors, those who have not adopted this technology, and in so doing, we are going to save even our sharing will be changed. Thank you so much for this organizers, and particularly I want to thank you even our partners, those who have fashioned this conference to be so much effective. We thank those who have joined with the NBA and the rest of the team, who have Carlo and the rest, and Kempis, who have joined us to come and do this, because indeed this is it. This is what we say, true food in due season. Because indeed, if we capture this, it definitely Kenya will change completely, and even the world at large will change. We thank you so much for these organizers, how they have done it, and the people who have participated, right from the farmers and the different scientists, those who have come to, they have given us their knowledge. This knowledge will not go to waste, but we are going to utilize it up to the end until we say Jesus comes back again. I don't know whether our speaker has come yet. Is he come?
He's just walking from the other side of the hotel coming.
Oh, okay.
You can continue to do
Okay.
2 minutes or so.
Okay. And this section, I want to ask if anybody who has experienced the same thing, just as I have talked about. I don't know whether somebody has a testimony like I have. Anybody? Okay.
You can recap what we learned yesterday. Sorry. You can just recap what we did yesterday.
Shall I remember?
Since you- Yeah, this one. We had
Yes, maybe we can, as our speaker, try to fix his gadget, we can go through what we learned yesterday. We had presentation by Fabian Raki about GMO, debate, science, politics, myth, and the fact and about GMOs. And again, we had a lot of knowledge from Josephat Mushiri about public engagement, capacity building, and biosafety communications in Kenya, GMO approvals, and all the processes. And then we had the opportunity to talk to Margaret Karembo of ISA about communication in biotechnological and biosafety spaces. Then we had a very touching experience by our daughter here, Evelyn Kimanthi, who really gave very passionate about the experience of the youth and young scientists. Actually, personally, I was so much touched by the talk of that young girl, because indeed, as parents, also we have not only scientists, elder scientists, we have a lot to play in bringing up our children so that they catch up and get they get the space that we are occupying, so that our youth can move to join together, and for this, our country can have a change as we have youth who are really in this issue of food safety. Then later on, we had the role of institutional of higher learning in biosafety and biotechnology by Professor Joel Ocheng. We thank you for those presenters, and without wasting time, our presenter today has come. Is on the with the front of us here, and we want to welcome our Nyamongo,
Yeah.
Dr. Josesta Nyamongo of Karo. Welcome. And we have our language translator. Is it Wanjiro? Yes, Wanjiko. Thank you.
Assalamu alaikum. Sorry for coming late. Had a small technical hitch with my car this morning. And when ilionekana kama nitachelewa, nikapiga simu nikasema nitachelewa kidogo. And indeed I'm late, my apologies. So I'm here to present to you the strategies for conserving genetic resources. My name is Dr. Nyamongo, as you have been told. I work with CALRO. I'm based at the Genetic Resources Research Institute. I will tell our story as we walk along. Now then, perhaps before I go into the strategies, we want to understand why conserve biodiversity or genetic resources for that matter. It is from those resources that we are able to impart production resilience to our varieties, especially now in the face of climate change, where we are getting several challenges. So breeders will look for resilient novel genes and insert them in the improved varieties so that to impart production resilience. Now, biodiversity in general, we have, they give us the ecosystem services. We are aware of the pollinators. They are organisms that assist us in nutrient recycling. And they are responsible for ecological sustainability. Now, again, for those of us who believe in God, the organisms are interdependent. If you go to your primary level biology, you was told they are producers, the food chain. From plants who are the basic producers to the highest animals who are the consumers. Food and nutritional security. The biodiversity itself ensures that there's food and nutritional security. Actually, the current findings is that even within the same species, there is diversity in terms of nutritional content. If you are talking, for example, for amaranth, there will be amaranth with more iron than others. The genotypes that is higher in, say, iron content. If we are talking about those ones that assist us protect ourselves against diseases, we did an analysis of Bambara nuts, and within the Bambara nut diversity, there are those genotypes that had a higher, they have anti-cancer properties, and there are those genotypes that were better in providing those anti-cancer properties than others. So that security, you require that diversity, and that's why, I mean, having one variety of Bambara nut is not good enough. Environmental quality and then economic development. So now let me tell the story about conserving genetic resources in Kenya. I've indicated that I'm from Gary. We were established by the Cultural Act 2014, as one of the 16, now 17 semi-autonomous institutes of culture. Our mandate is to conserve plant, animal, and microbial genetic resources in Kenya. The official long-term genetic resources is housed within the Gary Institute. That facility was initially established way back in 1988 with support, technical and financial support from the then Federal Republic of Germany through GTZ, now GIZ. At the other mandate is that we have both regional and global mandates. There are crops for which we conserve for the whole globe, for the whole world. For example, sesame, we are a global repository for sesame. We are an African regional repository for finger millet, pigeon peas, and cowpeas. And then we also conserve the national diversity. Why conserve genetic resources then? I think I've captured that, most of that in my first slide. They provide livestock feed, crop improvement, livestock improvement, extract medicine from them, food and infrastructure. That's why we conserve them, but if they could still provide that if there were no pressures. But now, most importantly, is that there are pressures that are eroding that diversity. The deforestation, the overgrazing, advances in agriculture. Now, as new varieties are introduced into the farming system, then farmers tend to neglect or abandon what they used to have. Their land races, because the improved varieties are more resilient or they're more productive. So the land races that produce less will be abandoned. So, and yet, from those land races that we want to extract the genetic resources that we will need to utilize for further improvement. The pollution which is having a devastating effect, the recent floods, which we have suffered from, they do not only affect mankind, but also the rest of the biodiversity. Population pressure, I'm not sure whether it's a good reason, because I think that we have been using that as an excuse. I think with the growing population, we should reorganize ourselves better and know how to live with biodiversity without impacting negatively on the biodiversity. There are two global regimes that govern the exchange of genetic resources. The CPD, that's the Convention on Biological Diversity, which has two supplementary regimes, one under which this organization has been established, that's the Cartagena Protocol, and it is about movement of genetically modified organisms. And then the Nagoya Protocol, which governs all the exchange of genetic resources in general. And now for food and agriculture, there is a specific regime that addresses the governance of how those genetic resources can be shared between countries, among countries, because we are all interdependent. The maize that we pride in as our staple food never originated here. It is originated in South America, Mexico. So if we are looking for novel genes for maize, chances are that they will be found in Mexico. That's how interdependent we are. And that treaty has a multilateral system which details out how countries or scientists can be able to exchange genetic resources. Both regimes subscribe to 3 objectives of biodiversity conservation. That is conservation, ensuring that the materials are there for posterity, sustainable use, and then exchanging of benefits, sharing of benefits that accrue from the utilization of those genetic resources. Now, the different components of genetic resources, as I've indicated, and we will use different approaches or strategies to conserve them. The different components largely include plants, plant genetic resources, animal genetic resources, insects and microbes, the aquatic genetic resources. We can largely group them into those components. Now, here specifically to talk about the strategies for conserving those plant genetic resources. Now, you can either conserve them as seed in a seed bank. You dry them to, you collect the seed, dry it to very low moisture content, 5% moisture content, and then deep freeze them under -20 in freezers. But that approach is only applicable for genetic resources that can withstand that kind of treatment. Not all seeds can withstand desiccation to that low level of moisture content. There are those seeds. There are those seeds that can, if you dry them, the embryo dies. The mango seed, for example, the avocado seed, if you dry it to that level of moisture content, it will die. So for those seeds that for those species that produce seeds that are not orthodox, which cannot withstand desiccation and the freezing to very low moisture content, then you have to employ a different approach for conserving it. These ones, they are either conserved in fields as living field gene banks, your collection of different ecotypes, you grow them in the field and you keep on renewing it, which is a little bit challenging, especially when you take into the cost of maintaining those fields and you have to regenerate from time to time. That's the same thing that is done for those species that do not produce the orthodox seeds that are vegetative propagated. Now, the other approach that you can employ is in vitro conservation. There are various forms of in vitro conservation where you grow them either as plantlets, tissue culture plantlets, and then work around with the medium in such a way that that micropropagation normally enhances the rate of production of the seedlings, but you can manipulate the media and the temperatures so that you slow the rate of growth so that you do not regenerate from time to time and conserve them as plantlets. If you have better capacity, then you can use cryopreservation, whereby either you extract the cell or the DNA or the pollen, the embryo, and cryopreserve it in ultra low temperatures. The nitrogen, the liquid nitrogen temperature, which is very low, that's the cryopreservation. So you have to develop protocols for addressing each of what you are dealing with, because different species, different components will respond differently to those treatments. So you do it in, you conserve them as cryopreserved material. Then, especially for land races, then what we have lately been doing is to conserve them with farmers in what we refer to as community seed banks. The other natural thing to do, especially for the wild foods and wild relatives of crops, you can conserve them in situ, especially in protected areas. An example here is wild sorghum or wild rice. We have about 6 species of wild rice in Kenya, and these are conserved in situ in protected areas. Now, where are we in terms of conservation in the country? We have close to, at the gene bank, we now have amassed close to 50,000 accessions, representing close to 2,000 plant species. Kenya as a country, we have about 7,500 plant species. There are a few that are yet to be described. There could be much more plant species that, just like other organisms that are yet to be described. But the current records indicate that's where we are. So if I'm saying we have conserved close to 2,000, then we are less than a third in terms of covering the country's planted diversity. That is the distribution in terms of the families that we have been able to conserve. The largest collection that we hold is in the grass family, and that is because of the pastures, especially the native pastures that we conserve. Then the legume family, the fabaceae, again, that is a majority of them again are forage legumes, but that includes the crop species that are either legumes or grass, including finger millet, for example. That's generally the distribution in terms of composition. In terms of the top species, our highest collection is sorghum, and this is because of being a regional repository for that particular species, followed by Avena, courtesy of the project that we had early in independence with countries like Canada, where much of that Avena was collected. Then the beans, the Phaseolus eleusin, and so they rank in that category, those categories. In terms of genetic status, I mean, about 12,000 of them are land races, 4,000 are wild species, 1,000, close to 2,000 are the advanced cultivars. We also keep breeders' lines for breeders. Now there are those that have been collected and they are non-classified. As they were brought in, it wasn't specified what kind of genetic status they belong to. They are the majority. What are the genetic resources conservation gaps? We are still inadequate in terms of covering the plant diversity. Now, I've already indicated that our main capacity is in seed banking. Remember the different options I gave you earlier that you can conserve. Our capacity at Igeri is much more in seed banking. These other approaches, we are yet to develop the capacity to engage in those other options. So because of that, there is quite a diverse material which cannot conserve as seed, whose species don't bear orthodox seed. They are yet to be covered. We haven't done very well in terms of animal genetic resources again. CAGRIC, our sister organization, does conserve some animal germplasm, But this is for elite breeds. Our local indigenous diversity has not been captured in those collections. A few are in animal studs with the Ministry of Livestock, but again, we are yet to capture the full representative. Microbial genetic resources, we are yet to venture into that. Insect genetic resources, not yet. Aquatic, I'm not sure whether there's anything that has been done. The other major gap is since our establishment, we have been generally been heavy donor dependent. Say that if there's no donor support, then all that we can do is maintain our collection. Generally, there are no adequate resources to expand our collection base, expand the capacity. The danger with that is that then we drive the donor agenda. If a donor comes in and tells you wants you to support in collecting sorghum land races, it's because he's interested in those land races. And through that project, then he will gain access and move away with our genetic material. We are not able now to negotiate properly the terms of benefit sharing because the donors views the project, however small it is, as being part of the benefit that they has already brought. Thank you.
Thank you, Professor Nyamongo, for that good presentation. In fact, you have shown that you have clearly given us that we have food. We have those genetic materials conserved for our researchers and for our development to some other scientific fields. Thank you so much. Now we have to shift a bit of our session. We had indicated that we have a presentation by a farmer, Mr. Daniel Magondu, but remember he did this yesterday. So instead of that, now we have to go another session of our panelists. Sorry. There are those who maybe have some questions. I think when he's seated here on the stage, you can raise some questions. Do you have mic around?
Thank you. Thank you very much, Facilitate, and thank you, Dr. Nyamongo, for making it. We truly appreciate that you have been able to come here. I have two questions. Number one, how do you respond to the claim that adoption of GM technology will erode our traditional genetic resources? How would you respond to that claim that adoption of GM or modern biotechnology will take away or erode or destroy or finish our traditional genetic resources? Number two, just for the sake of people who are here from the media and other practitioners who are not scientists like yourself, describe briefly what you refer to as in vitro and in vivo, so that people can get to understand what that means. Thank you.
Sorry, any other question? Before you respond, let's have another question.
Thank you, Dr. Nyamongo. It's always a pleasure to hear you talk about conservation of our genetic resources, and there's always more to learn from you. One of the questions I want to ask, a lot of the pressure to increase our food security is leading us to develop biotech products, particularly issues around insect resistance and also virus resistant. I'm wondering, in the collections that we have, the land races that we have, maybe of maize, do you characterize the land races to see what quality, what ability they have, and perhaps in your collection, do you have, maize or land races that maybe are resilient or have properties like resilience to diseases like MNLD or pest pressure from fall armyworm? Perhaps it would be good to know what do you know about those land races that you have? Maybe give us a few examples so that we are not rushing to adapt a technology and yet we have the solutions in our land races. I also want to know, because you said that there is a lot of donor, Gary is very donor dependent. I had the opportunity of visiting the seed bank, the seed conservation in Ethiopia, and their gene bank, and they told us that their government funds their gene bank 95%. They are only funded 5% by donors. What's the situation in Kenya? Thank you.
Do we have any other question? Yes, there's one here.
Thank you. I'm Tabby from Seed Savers Network. And maybe I can just start by appreciating the work that Gary is doing. We have worked collaboratively with Gary to promote the conservation of these indigenous seeds and also setting up various seed banks across the country. These are community seed banks that are owned by communities, and they're preserving their seeds in those seed banks. So my question is, currently 80% of the food that is produced in Kenya comes from smallholder farmers who do not employ maybe the technologies, who do not use seeds that are employing some of the technologies that are already existent and are using farmer-managed seeds. Maybe in your view, do you think that there is need to employ this? Like, do you think enough food is being produced? And maybe, since these sons are the ones who are already producing most of the food, that more should be done to enhance or to build their capacity in the efforts that they're already doing using indigenous seeds? Or should we start looking for other solutions that maybe they don't even need?
Thank you. Do we have any other question before he answers those three? Look like there is none. You can respond to those questions.
Thank you. Thank you. Does would GMO erode our diversity? Yes and no. Yes, if we adopt technology, the GMO technologies that are not. Let me go straight to it. There is a technology, and it became very famous, a GMO technology, but it never left the lab. And that has sent a lot of fear amongst the population globally that GMOs are generally bad. That was, in my view, as a conservationist, that was a very bad technology, the Terminator gene technology. If this was allowed to get out of the lab, Daktari, then that could definitely erode, because what it meant that you cannot use the next generation of the seed. the effect of that technology. So if it interacted, it cross-pollinated with our land races and spread that attribute to the land races, then that will be the source of eroding the diversity. But I want to assure the media and the scientists and the farmers who are in this forum that that technology never left the lab. So that's that's my first approach to it. Number two, before a GMO comes out, I'm sure maybe somebody who is a better expert than me will have explained the processes, the containment that it goes through, the tests that are done to see how it is going to interact with the local land races and land types in terms of perhaps transferring what may not be appropriate, the attributes that may not be appropriate. For example, if you are working on maize, on sorghum, this country has a lot of species of wild sorghum, all right? So one of the requirements is to show that, to make sure that when you are doing that, especially when you are doing your research and developing the variety that you want to develop, you do it in such a way that it is in a containment manner, that the pollen from what you are working on does not stray to either the land races of sorghum or the wild sorghum. So there are procedures that have been put in place to contain that. Now, if you do that without following those detailed guidelines, then yes, you are likely to erode the genetic diversity. But this country has procedures for developing those GMOs. Now, once a GMO has, if it's released, eventually released to the family, to the farming system, and yes, it's grown close to the land races, they will exchange the pollen. Is that bad or good? Under normal circumstances, in terms of enhancing the diversity, exchange of that pollen, the cross-pollination, is what grows the diversity. So if the GMO has been released into the farming system because of a good attribute, and that's what will make it, say it is resilient to drought, it's resilient, so it's going to impart that attribute to the land race. The fear should be, before we know what gains are we going to get from this GMO, you really need to contain the cross-pollination so that it doesn't mess up just in case. But if eventually the authorities will have released that GMO into the farming system for a good attribute, then it will exchange the pollen, and eventually your land race will have that good attribute. Whether that's good or bad, now that's not for Nyamongo to judge. Now, in vitro and in vivo. In vitro is what you do in the glass tube, in the lab, like the tissue culture plants. That is in vitro propagation. You are not doing it naturally from where you are not propagating naturally in the soil, in which what then it would mean you are producing in situ, where it naturally occurs. So if you have taken it from where it does and you are doing it in the lab, in the test tube. You are conserving that material in the test tube. Then that is what we refer as in vitro. Now, in vivo is in you. Let me give the layman language. You know, if you have food, you can eat it and stay long for not feeling hungry, so you will have conserved that food in vivo, inside yourself. In vivo is inside you. When you are doing experiments inside an animal, you are doing it in vivo within that animal, all right? So the major difference here is if you are anything in vivo is within the organism, and in vitro is what you the trials you are doing in the lab. If you are conserving in vitro, if you are conserving in situ, then you are conserving that material where it naturally occurs. Then there are different ex situ forms, which is now includes in vitro. Now, in terms of germ plasm conservation, I'm not sure the term in vivo will apply, but it will apply for other trials, like when an animal nutritionist is trying to know what kind of nutrients a given feed has, and is monitoring that. Those of you who have visited those animals and they have been slit somewhere in their tummy, and the scientist has inserted a bag there, and those materials are being digested within the animal. That is in vivo. I don't know what that assists to. Now, the characterization of our materials. There are those materials that we have characterized and we know their attributes. Actually, some of the materials we conserve have, for example, recently, there is a millet variety that was released under the national system of relating varieties. It was one of our accessions acquired from the gene bank, and it was through that characterization and evaluation, and it passed the tests for example, blast in millet. It was found to be blast tolerant, and it is for that attribute. So there are those ones that we have characterized. The majority of them we are yet to characterize them. So we have them, yes. We do not know their utilization potential because we have not managed to characterize them, and that's because of inadequate resources. Funding, I think in the whole of Africa, I don't know to what extent South Africa, but the only country in this continent that has gone full blast in terms of ensuring, supporting the conservation of their genetic resources is Ethiopia. I don't know whether it's because Ethiopia is the home of several crops, and that's that country is an origin of quite a number of crops. There's a lot of diversity. I attribute it to this factor that a former director of the then Ethiopia Gene Bank went into politics and became a minister. And things changed for that institution since then. He assisted, explained the policy makers the importance of their genetic resources. That's how I think I can only attribute it to that, the historical background, because at the Ethiopia Gene Bank, we were together, they are about 5 to 10 years older than us. It was established with support from Germany. We were established around the same time. But now they are strides ahead. That facility is a whole organization in itself. It's not like ourselves. We are part of CALRO. That one is an independent institution, and indeed, more than 90% of their operations is supported by the Exchequer. All our other African countries, including Kenya, the story is different. When the politics of genetic resources and biodiversity started in the early 90s, started in 92, when CPD started being negotiated, Africa, Asia, except Japan, and the whole of South America were in one side of the negotiation. We were the resource-rich regions. The North, which is now the Americas, America and Europe, they were on the other side. We were negotiating across the table with those as those two regions. The region with the resources, the region is with technologies and the funding. But since then, most of Asia has moved on. Korea, who were with us, they have moved to the north. In terms of, not the development for that matter, but in terms of how they look at genetic resources of their country. From South America, Brazil has since moved. They support most of the countries from South America. And India has moved on. China moved even earlier. China was with us. But Africa, I represent the country in those negotiations. I think sometimes I pity myself, keeping on that, saying in Africa we need capacity. Those countries that were also saying the same story are not telling the story this time around. Now, there was a last one from there, support. Indeed, those small-scale farmers need to be supported, and there are processes in place now to support them. You remember about, you might know about the review of the national city policy to accommodate the farmer managed city system. There are regulations we are putting in place to address the same. We did the amendment to the Seed and Plant Varieties Act to bring that system into board and inject some quality into it. Thank you.
Thank you very much, Dr. Nyamongo, for that effective answers to those questions. I think now we have come to the end of that session. We want to call upon journalists. We have to go to another session of panelists, journalists to come over and take us to the next level.
Good morning, ladies and gentlemen. Good morning. Hey, you people, you go very quiet. It's morning hours, and like I told you last time, today we're extending all the way to 3. So, my name is Peter Theuri. I'm a scientist in charge of what I'm talking about today. In my capacity is South African politics and the elections, but I will be having the real scientist with me on stage shortly. What I ask of you is to be patient in our 3 hours on stage. We'll try as much as possible, keep it very short and relevant. Sorry about that. So, I am very proud to be moderating the last panel, as I did the first one. And so you will allow me to eventually make the closing remarks, CEO. But before that, I want to invite my panel onto stage. Please join me in welcoming each one of them as I name them, as I call their names out. First, we have Professor Stephen Runo from Kenyatta University. Professor Runo is an associate professor and researcher at Kenyatta University whose research focuses on using modern tools in molecular biology to overcome biotic and abiotic constraints of food production and enhance the understanding of modern biotechnology, especially genome editing technologies. Karibu, Prof. Next, I have a panelist I've been with probably in every panel, Dr. Martin Mwirigi from the Kenya Agricultural and Livestock Research Organization, KALRO. Martin Mwirigi is a director of the Biotechnology Research Institute and a research scientist at KALRO. Karibu sana, Daktari. And finally, I have Dr. Paul Chege from ISA Afri Center. Now, Dr. Chege is a crop scientist with a bias in cereals breeding and biotechnology. He's a senior program associate at ISA Afri Center and coordinator of the Program for Biosafety Systems in Kenya, PBS. Every time I get late to the office, it's Dr. Chege who reprimands me. And he's last, that's why I called him last on stage, so he will be late. Karibu sana, Daktari. Great to have you, gentlemen. Now, as you see from our program, our topic today is on biotechnological interventions in curbing climate change. We had Mr. Magondo speaking very passionately about climate change and what to expect, and what to expect as a farmer, but where are the scientists on this and where is biotech on this? But first and foremost, before we talk about the intricacies of biotechnology, we will introduce what climate change is. I'll start with Dr. Martin. What is climate change?
Thank you, Bwana Theri, and good morning to all of us. I think that's an interesting question because the terms are very straightforward, climate and change. So which means there's something that is happening within our climate sphere, and that has to do with change. And this change can be attributed to the change in terms of patterns. There is what we knew as the normal climatical conditions, which were quite predictable. But currently we are in a sphere where the climate, the patterns are no longer consistent. Yeah, and this has been noticed because for the last 200 years, since we started doing a lot of industrialization, there has been a significant change even in the average atmospheric temperature. Actually, it has increased by around 50%. And that is majorly due to production of CO2, or what we call carbon dioxide, and methane. Those are the two main gases that interfere or control the atmospheric temperature. Just to give an idea of what happens in terms of science is materials normally, once they encounter electromagnetic energy, they vibrate and produce energy. The electromagnetic energy that comes on Earth comes from the sun. And that is what we normally call the solar energy. And the solar energy comes in form of wavelength. And in that wavelength, you basically have a spectrum that covers from X-rays, gamma rays, the visible light, like the visible, why you are able to see today is because your eyes can be able to experience the visible spectrum. And if I can illustrate, the Uri has a black tie, and what it means is the energy that is coming from the sun for the visible spectrum, all of it is being absorbed by that material. And that's why it becomes dark black. But if you If the material is reflective of all the low spectrum, that becomes white. Yeah. So that's the principle behind what you can see in different colors. Then from there you have what we call infrared. And infrared is where carbon dioxide and methane comes into play, because those molecules are able to absorb that energy wavelength and in essence vibrate. For you to understand how vibration works, we have the microwave. Microwave is also another sphere, a spectrum within the electromagnetic spectrum. So the way your microwave works is the microwave is able to interact with water molecules. Once it interacts with the water molecules, it produces heat. And it is the heat produced by the water that warms your food. So even in the atmosphere, it is the same thing that is happening. The CO2 and methane are absorbing the electromagnetic energy from the sun and basically producing the heat. Yeah. So the higher concentration of CO2 or methane you have in the atmosphere, it means more heat and in essence a higher temperature. Yeah. So for when we have increased significantly our temperature, what happens is the patterns within the globe changes. Because we increase the temperature, the glaciers within the temperate regions start melting. When it comes to the conventional rainfalls, the way we understand them, the more you raise the temperature, the more they become unpredictable because they are causing change of flow of wind because of that change of temperatures within the regions. Yeah. And in essence, you have now patterns that have either excess rainfall or no rainfall at all, depending on how the globe is responding. So the main contributor has been the production from industry, producing CO2. Sometimes we blame the animals, especially the cows, because they produce methane. And CO2 is removed from the atmosphere basically by the plants. The plants take in CO2 to manufacture their food. That is where carbohydrate is generated. The C, the CO2, the C is a carbon. And sugar is composed of carbon molecules. So basically the plant does is get the CO2 from atmosphere, and using the light also as an activator, it's able to make its food. This methane basically has no direct removal, but it has a lifespan of around 12 years because over time it interacts in the atmosphere and eventually converted to CO2 and removed from from the system. So basically the lifespan of a methane gas is around 12 years. Yeah. So for us to deal with now the climate change, then it means we have to moderate the amount of CO2 and methane within the atmosphere. So in simple terms, in terms of science, that is what is happening. So there's an interplay between the energy from the sun, the gases that we have, and the activities we are doing here on Earth. So if we continue without mitigating, chances are we will cook ourselves. Back to you. Thank you.
Now, I should have warned you that my panelists can give you master classes in physics, in mathematics, and everything. That was a class in spectral reflectivity, and I'm impressed with that. Thank you very much, Dr. Eri. Now, Prof, to you, because it has been made, it's now clear that the crops cannot grow the same way they used to because of this climate change. How is this biotechnology contributing to development of climate resilient crops and to enhance agricultural sustainability?
Thank you, Theuri, and really great to see everybody here, and well charged for until the last day of the workshop of the conference. So that's a really good question, Theuri, and coming from what Martin has described about climate change. So it's true that there is changes in weather patterns, and this will affect agricultural production or food sustainability in different ways. And there's two ways that I look at this. So the direct effect of climate change would involve increased temperatures, which means that there is drought. Plants don't have as much water as they should. And if plants don't have water, one of the consequences is that they don't produce as they should, and oftentimes they die. So there is mitigation against drought, and now we have toolkits that we can be able to use to mitigate against that. And then the other effect of climate change is biotic. So changes in weather patterns, climate change will lead or has led to increase in cases of pathogens. So we have a lot of infestations because of viruses. We have infestations because of fungal, as well as bacteria infections, and even on plants that I work on called parasitic plants. So in addition to drought effects, climate change will also bring about increase in disease and pest infestations. So cycle back to how biotechnology and gene editing, for example, can be used to elevate or to mitigate, to mitigate against the effects of climate change. And I want to be as simple as possible. One of the ways that we can use genome editing, for example, or biotechnology in general, in mitigating against drought, for example, is to reduce the rate at which plants lose water by regulating stomatal opening and closure. So you all remember your basic biology, the organs in a plant, or the components of a plant that allow loss of water or that cause water loss. So the stomatas. And there's ways that you can regulate the opening and closing of stomatas. And in effect, you'll have controlled water loss and mitigated against drought and in general climate change. So the other effect of climate change that I want to give as an example and leading to the consequence of drought is loss of chlorophyll. So chlorophyll is the green color that you see in plants, and that green color is supposed to be used for making food in plants, so photosynthesis. And one of the effects of drought is reduction in photosynthesis and reduction in chlorophyll content. But we now have tools, and there are plants and proof of concept works that have shown that you can actually increase chlorophyll in plants. Okay, so you can be able to regulate and increase chlorophyll, and in effect, you'll have mitigated against climate change. And there are many, many other examples. One of the key enzymes that is involved in abiotic stress or drought response is called abscisic acid, or ABA. And again, with genome editing, and I know you have had lectures upon lectures of what genome editing is, you can be able to regulate ABA so that it's able to deal better with regulation of drought and abiotic stress response. And then the last one that I want to point out is how plants would be dealing with drought effects. One of the things that happen is accumulation of reactive oxygen species, reed poison. And one way that you can be able to mitigate against that is to reduce, against drought, is to reduce the effect or the accumulation of reactive oxygen species. And we have tools that can be able to deal with that. Then going back to the side of biotic stress, which is more of where I work on, one of the effects of drought, one of the effects of climate change is increase in pest infestation. And one of the ways that you can be able to mitigate against crop pest infestation is using genome editing. And I want to use a live example here, and I don't want to use my example or the example that I'm working on. Calro, together with CIMIT, have developed MLND resistant or tolerant maize, and this has been developed using genome editing. And so for mitigating against the effects of climate change that lead to virus infestations or crop diseases, then a really good example and a case in point here is what Calro and James Karanja together with CIMIT are doing to develop MLN resistant or tolerant maize. And you'll bear witness with me, and you'll agree with me that it is true that climate change has led to increases in pests and diseases, and that we have tools using that as an example to control this.
I find it how impressive how Prof makes it sound simple. That sounds like work from tens of years, but how he says it sounds like something that can be done one afternoon. Coming to you, Dr. Paul. Now, through all this, there has to be regulations. There has to be a way to get to society what has been tested and what is, to acceptable standards. So what roles is our Biosafety Authority playing in the mitigation of climate change throughout the research that has been talked about?
Yeah, thank you so much, Peter. You said sometimes I'm your boss, so I'm trying to remain that even in this panel. but just to thank NBA for giving me this opportunity to even be in a panel with my seniors. As you have a senior, I also have my seniors, and they are right here. So I have learned from the best, as you can see. But going to the question that has been asked, and as we are told about climate change, and I like how Dr. Mwirigi has broken it down. I believe now you understand where we are and why it matters. Right now, we are being called by the Convention on Biological Diversity, one of the organizations in the United Nations Environmental Program, that is a custodian of the Cartagena Protocol on Biodiversity, which we have been talking about the entire week, and I believe now it's you can say it to the next person as if you are just describing water. So what we are being called to do by the Convention on Biological Diversity since the last meeting of the parties in Montreal in December 2022 is to be part of the plan. And on May 22, we celebrated the International Day of Biodiversity. So what that means, or the plan that CBD is talking about, is a plan to halt and reverse biodiversity loss. And they have targets. They are calling the Montreal Global Biodiversity Framework. These targets are about 23 in number, and there is a part, there is your part. Whatever you do, there is your part in those targets. I encourage you to look at them when you retire. But coming to NBA and its role, NBA is the focal point in this country for the Cartagena Protocol on Biodiversity, and so they represent us in the international negotiations on these targets, on the framework, on the global framework. And NBA, what we expect them to do is to, because I don't work for NBA, so I will not say what they are doing, but what we expect them to do is to be an enabler of technologies that halt and reverse the effects of, biodiversity loss due to the effects of climate change. And as Prof has explained, there are many reasons that biodiversity can be lost. And I want just to say that, to add on what he said, sometimes we are also witnessing increase in invasive and migratory pests just because of climate change. They are looking for better environments to thrive, and that's why we are witnessing fall armyworm, which we didn't used to have in the last couple of years. So what we are asking NBA to do is, as they continue to regulate the technologies that come with a promise to halt and reverse biodiversity loss, what we are asking them to do is to be guided by science and evidence as much as possible, so that the scientists that are doing their work are guided by science as much as possible. The people that are receiving it are also receiving the technologies based on the evidence that this is going to do exactly what it was meant to do. I also want to bring up something called sustainable intensification. Sustainable intensification is one of the other promises that these technologies promise to do. This is production of more food with less resources, and we have had that throughout this week. That what we are asking Prof to do as they develop their Striga weed-resistant sorghum is that the Striga, the sorghum that we have been producing gives us yield. The same sorghum, in the same plot, without the need to expand to another field, to open up another land. Let it give us yield right there, without the need to, and you know what happens with opening up new lands. You are interfering with the biodiversity. So if that can be done, we can get produce with the same resources that we used to have, with probably less water use. We obtain produce with less need to invest in insecticide sprays. We get the same produce. We will have made a stride. So we are asking NBA to be guided as much as possible by science and evidence.
Thank you. My boss, today I'm your boss, at least for the next 5 minutes. The next question I'm asking you is about football, so at least I can say I got one over you. But before I go to my follow-up questions, I'd like to throw back to the audience so I can get one or two questions for my panel. You can ask literally anything, even about South Africa. I'm here to answer the elections. And I know the last day of the conference is probably going to be more active than the days we've had, so shoot them.
Yeah, thank you, the panelists, for the great discussions. Maybe my question is out of curiosity. With the direction the country is moving on carbon markets, and Kenya has been identified as one of the countries with that can be used as a carbon sink. How can biotechnology be used to improve, to modify the genetic structure of these forests and these carbon sinks, just to increase the ability for carbon sequestration? Thank you.
Thank you for that very elaborate discussion. My question goes to Dr. Mwirigi. How can we moderate the carbon dioxide in the air?
We can take two more and then we get the responses before we pick another 4.
Thank you, panelists, for your discussion on climate change. We are aware that climate change is a big problem for Kenya and the world at large. Even though we are not emitters, we suffer the consequences and we have to adapt. And I think we must look at the low-hanging fruits as we look at what solutions to our challenges. One of the easiest things to do when we are trying to adapt is to use agroecology or organic farming and put back carbon in the soil. Why we have to develop a GMO to cope, to sequester carbon, I don't understand. If we can only encourage our farmers to do more organic farming and use compost and put back carbon, it's a good way of adapting. Not only will we increase our soil fertility, but we'll also be putting back carbon in the soil where it belongs. And if you look at the survey that was done by, I think it was Tegemeo Institute in 2014, soils that were tested throughout this country, in the counties that can grow maize, none of the soils met the requirement for increasing for maize productivity. None had enough organic matter, and you can check that the soil suitability test. None of the counties had enough carbon in their soil. So by us promoting putting back carbon in our soil, we not only mitigate, but we also, I just don't understand why we are trying to look for solutions, and we have low-hanging fruits we can use. That's one thing. And we can perhaps look at biotechnology to solve other problems. But for climate change, I am convinced we do not need GMOs to solve our climate change issues. To me, that's more of a comment. So let's put our energies elsewhere. Thank you.
One last one.
Mine is directed to Dr. Paul. You talked about the NBA functions or mandates. You said that NBA should be guided by the science and evidence. And also you talked about NBA, one of their mandates is to regulate the technologies of research. Is it, are you emphasizing that they should adapt that, or are you, mine is more on clarity, is NBA already doing that? Am I more of a suggested or emphasizing that they should adapt the mandates?
Okay, we take those ones first. Okay, one more.
I have a small question, probably out of curiosity. Actually, what is carbon trading? How is it done? And how do countries benefit? Thank you.
Great, let's answer those ones first. My senior.
Yeah, I think I will just respond to those ones on this side, and then I let my panelists tackle the rest. Thank you for the questions. I start with the last question about whether NBA has been already regulating guided by science and evidence. And to the most extent, I can say that is this the case. So NBA is already setting, trends in regulation based on science and evidence, despite the call to, reverse this based on sometimes persuasions, perceptions that probably are not backed by any science or evidence. So I urge them to keep up that, and let's have technologies that enable us to, produce, because we have to feed a population, a growing population. We have to protect our farmers. You saw the statistics, the grim statistics that the youth are not in agriculture anymore. We have to make agriculture attractive to the youth. That is not through hoe and slashers and panga, or what we used to call slash and burn technology. We want agriculture to be more attractive and more rewarding, and in the end, giving us sufficient food. We cannot do that without tapping into technology. That's why the government, in its wisdom, has put up, and I see the chairman of the NBA here, continue to fund NBA so that we regulate technologies that are produced by scientists. Otherwise, without the scientists, without the need for the scientists working, there wouldn't be NBA, we wouldn't be here. So let's give credit that NBA is doing its job. I wanted also to respond to the comment that came from our sister, Wanjiru Kamau. Tells you that even GMOs are cultivated organically. There is no reason why you shouldn't use manure in growing the cotton, in growing the GM crops. A GM crop, and we go back to all that we have learned this week, is just enhanced with a gene specifically for a certain purpose to maybe protect it against a menacing pest or a disease that cannot be controlled otherwise. Let us not see as if some technologies are preserved for some quarters. We as scientists actually promote even organic agriculture as well, because we understand the purpose of adding organic manure in a soil. It improves the microorganisms, it improves the soil water holding capacity, it actually improves the uptake of nutrients that are in the soil. All that purpose, we need that for GMOs. GMO is not a panacea. It will not solve all problems. We actually encourage farmers to also see, and you also don't use manure, without information. You use manure when advised by agronomists, by these same scientists. So I think you will still come back to us to advise you that soil that you are putting manure on probably don't even need it, because probably it has even more than enough microorganisms. Thank you.
Thank you, Paul. I think the hard questions will be left to the senior the senior people in this panel, and this is the seniority is going. Theory is junior to Paul, and Paul and I are the same. This is but I'm really, I like to be very pragmatic, and I like the questions that have been asked here. I'm very pragmatic person. Very evidence-based, and I like the question, especially on the sequestration of carbon. I'm not an expert in carbon sequestration. As a molecular biologist, I know possibilities are limitless. If there's a gene, it can be tweaked, and it can be changed to increase growth, to increase photosynthesis rate. It can be tweaked to make leaves broader. It can be tweaked to make leaves look like this. There is all sorts of possibilities. We are not short of those possibilities. But I want to be very pragmatic here and give a real example with a view that it might change some people's views here about how we can use biotechnology to really dramatically reduce carbon footprint. And I'm sure that is coming. Not a prophet, but I can foresee that happening in the next few years. So think about streetlights. Streetlights are really expensive to maintain, and that's probably why we don't have streetlights in most of our streets here in Nairobi. in Nairobi or in Kenya, because they're really, really expensive to maintain, and that draw a huge amount of energy. They emit a large amount of, they produce a lot of emissions. So the footprint or the energy that is just based on, say, streetlights alone is a lot. So think about if you can have trees on the streets. So think about our new expressway. When that expressway, instead of putting lights there, so at the bottom there, those pillars that I don't really like. So imagine those are trees, and those trees are emitting light, so they're illuminating the streets. And I said with molecular biology, as one, the possibilities are limitless. So one way that we can be able to do that, and there are already scientists experimenting with this, is to make the trees glow and emit their own light. So you'll have killed two birds with one stone. One, illuminating the streets, and then sequestering carbon. But not happening. I'm not a prophet. It's going to happen.
Yeah, thank you, Prof. Professors also do philosophy, and now they are, it's called what? You prophesy. There's philosophy, and now there is prophecy. So I think a number of questions have been asked, and I'm going to start with one on organic farming, just to re-emphasize what Paul has said. Organic farming is a system of a plant or crop production. Basically, when you say organic, it means you didn't use synthetic fertilizer, or you did not use chemicals to basically control diseases and pests. That is the meaning of organic farming. It has nothing to do with a crop or a plant. So you can grow a GMO organically. And actually, when you look at the design, like now we are talking about pest and disease resistance, it means you are no longer using the chemicals. So a GMO actually is more compliant for organic farming. Yeah. When it comes to maintaining the soil integrity, we are talking about herbicide tolerant. In other words, you'll use chemicals to just clear the weed. There is no tilling. And when you don't do any tilling, it means you maintain the soil integrity, and the ecology, the system down there is not disturbed. And you don't lose water. And you don't lose the carbon. So basically, the herbicide tolerant is an ideal, actually, for carbon sequestration in the soil, because you are not breaking the soil and you are not disturbing whatever is happening there. So a GMO that is designed for that is actually ideal. Yeah. So I hope you have that understanding. And then Bwana Magondo asked, how do we moderate the CO2 or the carbon in the atmosphere? Basically, there are two ways, because it's carbon, like Wanjiro said, is in the soil and also in the atmosphere. So when you do sequestration, it means you want it to be harvested somewhere. The main harvester for CO2 are the plants. So the more crop or the plants you have, the more you are removing the carbon from the atmosphere. The more you are not tilling the land, the more you are also not removing the CO2 or the carbon dioxide from the soil. And that comes to the carbon trading. This is an agreement globally, basically to try and mitigate the climate change. You see, in this world, you can either adapt or you can mitigate. The world wants to do both. In mitigation, you want to have more plants on the Earth's surface. On the other side, to adapt, you are basically creating animals or plants to be more tolerant. Like now, when you make a crop to be more tolerant, it means you are no longer affecting per se your target. is to be adapting to that increase in temperature, that drought. And in essence, when the crop, like he said, you can affect the chlorophyll, the structure, the physiology, but also you can influence, let's say, the maturity. Like the crop, the ones we do for adaptation, naturally, I mean, a conventional way for adaptation for drought is you make the plant mature early, so that even if it rains a very short period, you can be able to get some level of productivity. When it comes to adaptation also, maybe I can emphasize, we have our animals producing methane. When you produce methane, it means you're increasing the possibility of increasing the temperature. So when you look at one of the things to do is probably increase productivity. A Kenyan cow, dairy cow, produces an average of 7 kilograms. In Europe, the average is around 30. So which means in Africa, you need like 4-5 cows to produce where 1 cow in Europe is producing. Can we see the difference? And that's why we are being blamed, because we are having breeds that are many and the productivity is low. And that's why we need to be able to advance and increase our productivity so that we can have less animals, but giving us the same amount of product that we need. Yeah. So I think I almost forgot. Let me now explain the carbon, the dynamics that is there. The factories that are in Europe, in, let's say developed world, let me not use Europe, are still don't want to stop production of whatever they are producing so that they continue using the energy that is from the fossil and giving us the CO2 in the atmosphere. And since they don't want to stop, then we tell them, you can compensate by enhancing production or, planting of trees or vegetation that can actually be able to take up whatever you have produced. And since they don't have where to plant, Africa forms a good basis. So they come and tell you, for us, we have been charged a certain amount as a company to mitigate the production, the temperature increase due to the CO2. What level of acreage do you need to plant, either trees, so that you can be able to carbon sequester? And then they tell you, so that you also don't harvest that tree as fast, we are going to compensate you. And that is where now you are hearing about carbon trade. So there are companies being charged, the money is collected, but now they need to compensate you to maintain the vegetation or the trees that you have. And the dynamics are there. I think the government is working, has been working overdrive to have modalities. I think before there were no guiding policies. People were doing things the way they knew. But I think currently there is good guidance in terms of how the money should go to even the people who are maintaining the plants. Then I think that's all I can say for now. Thank you.
Thank you, physicist. Prof, back to you before the prophecy is gone. The gift of prophecy has disappeared.
It's inspiration.
How do you envision the role of genome editing now probably evolving over the next, say, 10, 15 years in context of the global climate change?
Yeah, I think this is changing really fast. Knowledge is exploding really, really fast. Again, I like to be very pragmatic. I do my analysis very based on data. Okay, so there's data to suggest that in the next foreseeable future, you will have maize commercialized, and that maize that is commercialized will be tolerant to maize lethal necrosis, one of the consequences of climate change. And this is happening right here across in Naivasha, I guess, where the trial is. So for sure, the next foreseeable future, so that would be the next, in the short term, that would be in the next 5 years. And there are many other crops in the pipeline. There are drought-tolerant maize that have been developed, probably not in Kenya, but elsewhere, that have gone through the proof of concept stage. Okay, so proof of concept just means that they have been developed and they have been evaluated and they have been found to have a certain amount of acceptable drought imparting result. And then again, closer home in Ethiopia, there are grain which is really resilient to drought. So teff is a really good crop that is resilient to, and together with other millets and sorghums and most of our indigenous crops here, most of those have already been developed, improved for drought tolerance, for example, and for increased yields to increase food sustainability. And those ones have, again, gone through the proof of concept stage. And I think in Ethiopia, they're just waiting for promulgation. That's a really difficult word to use, but you get what I mean when the former president Kibaki lifted the constitution. That just means that the rules in Ethiopia and the guidelines have come into effect. And once that happens, then we'll have teff that is lodge resistant, and that just means that we'll have a lot more food. And for me, those I'm calling the first wave or the first generation of specifically gene-edited crops that will be developed or that have been developed, and that will happen in the next 5 to 7 years in my prediction. And then you have the next generation or the second wave, which are products that are again under pipeline development. And I just want to point out one that is really inspiration to me, and because I think this is really excellent science and thus has a potential to greatly improve food sustainability, and especially in sub-Saharan Africa that is prone to adverse effects of climate change. So there is development of sorghum varieties, sorghum hybrids, and I'm using the word hybrid here to mean heterosis, which is the most technical word that I'm going to use today. Heterosis just means you have increased performance of a crop because you have increased heterozygosity, the other technical word. But what I just mean, hybrids are produced by crossing two varieties of crops, or two varieties of, or two genotypes, and the cross produces a seed that is very highly performing. And if you want to know the importance of hybrids or how hybrids can increase food production, just look back at the Green Revolution. The Green Revolution was spurred by hybrids. In Kenya today, if you want to produce a hybrid seed, you cannot. So that has to be the work of a seed company. So seed company X would have a way of mixing two maize lines to produce a sum of the parts that is greater than the individual parts apart. And when you plant that, you get really heterosis or hybrid vigor. You can only get hybrid vigor if you get that seed from that seed company. But with genome editing right now, you can be able to immortally produce hybrid seed. It's called apomixis. Apomixis is asexual reproduction of seeds. So that means you don't have to go to Kenya Seed, no offense, Kenya Seed, but I think soon we'll have to find alternative ways of earning money because hybrid seeds will be coming. And I think that's a really positive thing. And when you look at sorghum, for example, which is hard to produce seeds of, hybrid seeds of, and the fact that you can produce these seeds perpetually most generations, and a farmer would just plant and then replant that seed, and then replant that seed through as many as 7 generations without having to go to a seed company. I think that's positive, don't you think? And I see that coming in the next maybe 10 years for sure. Not a prophet again.
That was a big class. Prof, if I should follow you to K after this, I should get a certificate. I've learned enough. Before I get back to you, Dr. Paul, can I throw it back to you one more time in case we have questions, the final round of questions as we come back to the panelists, starting by my senior. If we have one more round of questions, anyone? That could mean that Prof was so clear with heterozygosity and everybody got it, or that he wasn't Prof and nobody got it. Okay, we'll assume you're good. Dr. Paul, I'll give you more minutes than I've given the other two because I fear to be punished. But as you conclude now, tell us what you think about the regulatory landscape and the expectations in future for Kenya. What better they can do than what's happening right now? What improvements can come? What you expect generally? And your closing remarks.
Yeah, thank you so much. Just before my final thoughts, I just wanted to say, because I probably will not have another chance, and since I'm here, to appreciate the presentation by Dr. Nyamongo on gene banks and the importance of preserving our genetic diversity. And also to correct thought that went around as questions were being asked, that scientists are diminishing genetic diversity by doing what they do. In fact, scientists are the biggest beneficiaries of genetic diversity. Professor Runo here will not go to his lab to begin his work before combing through genetic diversity that exists of sorghum in Africa. So he probably has quite a lot of that, a lot of data, specifically looking for resistance genes to a certain trait. So that if that is lacking in one of a farmer preferred variety, because farmers also have preferences and they also do selection. So what we work with as scientists is what farmers have actually advanced in selecting. So he looks at the profile of that farmer preferred variety and also looks at the genetic diversity that Dr. Nyamongo and his team has preserved over the years and see whether there is a resistance that can be borrowed there. So scientists are the biggest beneficiaries of gene banks. And I will tell you, because we have CGIR systems, we have 2 in Kenya, I think that was the main idea why CGIR systems were formed in the first place, so that they can preserve genes. So that is one. The other thing is that it is probably not cost effective to do genetic mapping or study the entire gene bank. I don't think it will be very wise. I think it is wiser if you have a challenge so that you can go to that gene bank and look for a solution. Otherwise, the land races that have been collected are in the millions in this in the gene banks that exist in the world, and you may not. It is not humanly possible to analyze all of them. So in my closing submission, I just want to urge the regulators and here NBA to be guided as much as possible by science and evidence and be, what do you call, what is the opposite of reactionary? Proactive in looking at emerging trends, emerging threats, emerging science, so that we are able to develop guidance for those in good time. We are able to make sure that our regulations work, not entirely be a policeman, but work with the population, so that you can be able to provide the food that is direly needed by our citizenry, which is growing every day. And here I will give an example of data transportability. Are we able to incorporate that? I know there is a clause in our law that allows us to borrow data. For instance, farmers that are devastated by Maruca pests, already there is a solution that exists. We have seen Nigeria and Ghana benefiting from varieties that are resistant to Maruca. So do we need to go to CFTs to generate data that already exists? So how can we move with the times, with the current trends? Thank you.
Professor Prophet.
Yeah, so as I think as we finish, I just want to reiterate one point here, that we don't do science for the sake of just fun and doing science. It takes a lot of effort, takes a lot of money, it takes a lot of sacrifice and thinking on our part. And what we try to do is to find solutions for some of the most, and for us here in Kenya, and for me in particular, is to find scientific solutions for some of the most intractable problems that we have here. Problems that nobody from, say, outside of Africa would be solving. And I think what we can do as African scientists and as Africans and as Kenyans here is to support those technologies. There are real solutions that can come from science. And like I said, it's not just for fun, it's for solving problems, and these problems would not be solved by anyone else except us. And if we can find those solutions in science, then why not? And the last point I want to emphasize, and this is true, and this has been said many, many times, the greatest resource that we have, especially in Kenya, better than most other countries in Africa, and this was said by none other than President Biden, is our human resource. So there are a lot of intelligent Kenyans here in this room, in our universities, in our regulatory authorities, that are happy and capable of using the intelligence to improve our food systems and mitigate against the effects of climate change. With that note, my senior.
Yes, senior.
Thank you. I think mine would just to say one underlying thing. As Wanjiru said, Madam Wanjiru, Africa is suffering the biggest in this climate change. We are not the main contributors, but we are the main sufferers. So we need to see how we thrive under that circumstance. So we can either mitigate or decide to adapt. If you decide to adapt, it will be like the case of the frog which fell in water that was being heated. I think some of us know. And adaptation means, yes, you will be thriving at the beginning as the water is warm, but the heat will continue. So the last thing that will happen, the water will cook the frog. So it might look a good thing to adapt, but mitigation would be the best thing. Because already scientifically we know what is happening. It's just a case of doing the right thing. And biotechnology actually offers the biggest, in this case, contribution in terms of that adaptation. Because for us to deal with the diseases and pests, which are actually being caused by the climate change. Because sometimes when you hear climate change, we think it's actually very far. Even here in Kenya, I can give you an example. The increase in temperature, now we are seeing mosquitoes where they were not. We have one mosquito which we call Culex, which is the one responsible for spread of Rift Valley fever. So the highlands seem to have been a good place for not having that disease. But we are now starting to see some level of evidence of viral circulation within areas that were not infested before. Yeah. So biotechnology comes in to help us mitigate so that we can be able to improve our vegetation cover, because if our plants, our crops are protected from the disease, chances are then we'll have a better vegetation. I can tell you the same diseases we see in our farm, they are the same you find in the forest. It's not that the forests are immune. And Kefri will tell you there are some vegetation they are losing because of diseases. And some of them can be mitigated through the use of biotechnology. Because what is happening is the new change, the new level of disease, pests, infestation, we can just agree the common method or the conventional methods we have might not be adequate to address. The new diseases like MLN, which is coming from combination of two viruses like we had the other day from Dr. Karanja, tells you it is true. Changes of temperature is leading to other things like those diseases. So the way we can maintain our crop is to make it resistant. Because I can tell you, low-scale production, you can use the conventional methods. But the truth is, for us to feed a population that is going beyond 50 million, small-scale way of production will not be sustainable. It's true we all want to eat kienyeji chicken, which I would like to promote. You can come to Karo for kienyeji chicken. But I can tell you, Nairobi in itself, you might not be able to provide enough kienyeji chicken to give that population. And that's why we have to go to other methods of higher productivity, more intensive farming, to be able to feed ourselves. Otherwise, we become reliant on other producers, and Kenya will be losing, because like it was said the other day, we are spending over 500 billion to import food. Whereas we have enough resources, we have water, we have what it takes, but because we haven't done the good thing that was said in terms of managing ourselves properly and also using the right varieties, we are in the state we are in. Other countries do not have the resources that we have, but basically are able to feed themselves. And an example is Egypt. Egypt is a one-river country, not two, just one. And their population is more than double ours. And their soil, they're even worse. They import the soil. So basically, for us as Kenyans, unless we sit down and agree on some of use of these technologies and become intentional, because one has to become intentional. And I can give you an example. Ethiopia was given an example in terms of conservation. Right now, Ethiopia, when you look at where we are in terms of biotechnology, they have overtaken us. 10 years ago, Kenya was like miles ahead. We were seeing ourselves like 20, 30 ahead of Ethiopia. But their government decided to become intentional and invest in it. Right now, they have state-of-art labs directly funded by the government. That being intentional and deciding you are going to feed your population. But if we continue debating and seeing whatever will come, what is not there, we prophesy. By the time we prophesy 10 years to come, we'll have lost a huge number in terms of even our population, just because of hunger. So for me, my plea is, let's look at biotechnology as an enabler, or even dealing with the climate change, and basically even feeding our population. Thank you very much.
from spectral reflectivity, electromagnetic spectrum, to heterozygosity and biotech prophecy, to data transportability and regulatory utopia, to mine, which is Malema for President 2024. This is my panel, Dr. Martin Mirigi from Calro, thank you very much, Professor Stephen Rono from Kenyatta University, Dr. Paul Chege from ISA Afri Center. Our sign language interpreter today was Rosemary Njuguna. Thank you very much. She has not been appreciated since day one, so let's appreciate her. Thank you very much, and help me in appreciating the rest of my panel. Thank you very much. We close today. I think we can, after this, we'll also have the courtesy of giving the final word, because I think we've nearly exhausted everything. Thank you very much. See you some other time. I wish you all the best.
Thank you very much, our panelists and our moderator. We are coming to the tail end of the conference. I am only here to do one thing, to invite the participants at the closing session. We have only two things to do. I'd like to hear from the team that was conducting the evaluation, whether that has been done. We filled in forms. So there are some forms that we're asking a few questions for purposes of feedback. Please fill that in and hand them over to our colleagues from the NBA. Those who are present in that team, I can see Frida. Frida, your hand up. Frida is there, can pick the questionnaire or the forms. We are basically looking for even the next year's themes. We get them from your feedback, and also next year's participation. It is after confirming that we have enough of that data, then it is my pleasure to invite our colleague, our director within the National Biosafety Authority, Dr. Mothe Gilbert, to do the closing. Mothe is doing the closing on behalf of the Principal Secretary, Dr. Paul Rono. Please put your hands together to welcome our chief guest at the closing of this conference. Karibu, Daktari.
Good afternoon. Is it afternoon or morning? Morning. Good morning. Yeah. Thank you very much, participants, for this opportunity. It was not really intended. I've been one of the participants since Tuesday. Unfortunately, I left because we have an exercise that is going on, that is exhibition, pre-Mandala Kande exhibitions in Bungoma. And actually, the ministry is leading because the theme for this Mandala Kande is food security and nutrition. So everybody is in Bungoma, especially Cream House is actually virtually empty in terms of technical staff. And I was requested to be taking care of the emergencies because of the proximity between Naivasha and Bungoma. Somehow, the principal secretary was to be here. He actually had planned in such a way that yesterday would be around, and today you will pass by, and then again leave for Bungoma for tomorrow. Unfortunately, there was some change in the program that made the whole team to leave Nairobi on, that was yesterday morning after the prayer and breakfast, to Bungoma. So I was requested to come and do the needful, and that is what exactly I've come to do. So bear with me, because probably you are expecting somebody else who is not part of NBA. But for today, I may be wearing an oversized speech by Dr. Paul Lono, Principal Secretary, Ministry of Agriculture and Livestock Development, during the 12th Annual Biosafety Conference at Sawel Lodges, Naivasha, that the 1st May of 2024. Professor Genesio Kenyamario, Chairman, National Biosafety Authority, Member of Parliament, Justice Kemae, Agriculture, Chief Executive, Dr. Loy Mugela, our keynote speaker, Professor Richard Oduo, John Komen, Director, Program for Biosafety Systems, Dr. Margaret Karembu, ISA, Africa Center, invited speakers, the media, board members present, distinguished participants, ladies and gentlemen, good morning. I am delighted to be with you at the conclusion of this 12th Annual Biosafety Conference. I want to extend my thanks to the National Biosafety Authority for arranging this conference, which has been a yearly event since 2012. I also want to acknowledge the collaboration with the foreign institutions in hosting the conference. The Program for Biosafety Systems, the International Service for Acquisition of Agri-Biotech Applications, Koteva Agri-Science, Bayer East Africa, National Agriculture Fund, National Research Fund, the Open Forum on Agricultural Biotechnology, the African Agricultural Technology Foundation, the United States Grain Council. The event would not have been successful without the participation of government parastatals present. To mention, National Housing Corporation, Kenya Team Board, KEFIS, CARO, as well as 9 ministries, including Ministry of Trade, Ministry of Devolution, Ministry of Health, Ministry of Agriculture, Prime Cabinet Secretary, Ministry of Youth, Ministry of Environment, and Ministry of Education. I also want to thank Mr. Daniel Mangondu, a cotton farmer from Kirinyaga, Madam Jundi Abog, the Chief Officer of Agriculture, Omabe County, member of Agriculture Committee in Parliament, and all the other participants who took the time to be part of this important event. Additionally, I appreciate the ISA Africa Center for sponsoring the 9 ministries to be part of this event. I extend special thanks to OFMB Kenya for organizing the luncheon that took place on the 2nd day of the conference. Ladies and gentlemen, as we wrap up this enlightening discussion on modern technology and biosafety within the contents of our bottom-up economic transformation agenda, I am filled with deep sense of hope and determination over the past 4 days, we have evolved into the potential of biotechnology to drive sustainable development while improving livelihoods. This has been made achievable through understanding the government bottom-up economic transformation agenda, the legal and regulatory framework for biosafety in Kenya, the biotechnology and biosafety in sustainable agricultural transformation, public engagement, capacity building, and biosafety communication, embracing modern biotechnologies. Furthermore, this conference reflects the country's vision to play a significant role in global biotechnology sector, as mandated by the authorities responsible to oversee the transfer, handling, and use of genetically modified organisms. Kenyans should therefore feel secure that an dedicated government agency exists to ensure the safety of biotechnology products before they are released to farmers and consumers. I note with appreciation the slides made by the authority. Some of them include development and publication of necessary biosafety regulations to support operational mandates of the authority, the operationalization of the GMO testing and molecular laboratory, the approval for commercialization of BT cotton with resistance to devastating cotton bollworm pests. The BT cotton is currently being cultivated in more than 20 counties in Western Kenya, Coastal Region, Eastern and Central regions. The approval for cultivation of BT cotton has started bearing fruits, with thousands of jobs already created at the farm level. And the value chain processes including aggregation, ginning, spinning, weaving and knitting, and garment manufacturing. The approval for environmental release of BT maize improved for resistance to maize stem borer pests and genetically modified cassava that is resistant to cassava brown streak virus, both of which are now in the final stages of variety evaluation towards commercialization. 4, the migration of NBA services to online platforms that's facilitating quick and expedited processes of application. 5, decentralization of NBA services to key entry points of the country. And finally, 6, acquisition of prime land that will host headquarters and, more importantly, a demonstration field to showcase approved biotechnology crops to farmers, among other achievements. Ladies and gentlemen, being cognizant of challenges faced by the regulator, I call upon authority to explore innovative ways to mobilize resources for enhancement of its program on public education and awareness on biosafety. Therefore, accelerating adoption of safe technologies and contribution to food security. As a ministry, we undertake to continually support the authority to effectively discharge its mandate. This conference has presented the perfect platform and opportunity to explore the potential for the application of biotechnology as a tool for accelerating economic transformation. Finally, ladies and gentlemen, as we plan for the future, let's be guided by a common vision of a country where biotechnology acts as a driving force for grassroots economic change, where innovation is utilized for the greater good, and where everyone is included. Let's work together to create a more prosperous, sustainable, and inclusive country for future generations. In closing, I extend my heartfelt appreciation to the National Biosafety Authority for organizing this 12th conference. All participants, stakeholders, and partners who have contributed to this dialogue, your insights, expertise, and unwavering commitment are invariable as we strive to realize our collective aspirations for a better tomorrow. Thank you, and God bless you. And now, ladies and gentlemen, I want to declare this conference officially closed. Thank you very much.
Asante sana. Thank you very much, Director. We truly appreciate. We will ask our media team that we do the first stanza of the national anthem in English, and the Kenya national anthem and the East Africa national anthem as a way of closing and as a way of Thanksgiving prayer. If you can find that clip, we will be happy to do that. We will do that while standing in respect and honor of the national and East Africa anthem.
Afrika Mashariki, tuwezeshe kuishi kwa amani, tutimize na malengo yetu. Jumuiya yetu sote tuilinde, tuajibike, tuimarike. Umoja wetu ni nguzo yetu, idumu jumuiya yetu.
Thank you. God bless you. Have your going home in your returning to your residence. We welcome you for a cup of tea, networking session. We have also prepared lunch. Do not be in a hurry. A cup of tea, networking, lunch, and then you can take to your journeys back to your destinations. Asanteni sana na Mungu abariki.